In today's video, we will discuss about the prokaryotic cells. We know that all living organisms are made up of one or more cells, and the cell is called as a structural and functional unit of life. The cell theory was proposed by Sladen and Schwann, and according to them, all the plants and all the animals, they are made up of one or more cells. Similarly, they stated that the appearance of the organism and the functioning of the organism is a result of the aggregate action and organization of the cells present in their body. And the third postulate later on added to this cell theory, which explains how the cells are produced. So third statement was proposed by Rudolf Virchow, and according to him, omnicellula e cellula, this is the statement, which means that each cell arises from pre-existing cell. So cells, they multiply into daughter cells and number of cells, and they form their body. We know that during fertilization, the male and female gamete fuse together. It produces the zygote, single diploid cell. And later on, this zygote divides and redivides to produce the embryo. It differentiated into radicle and plumule in case of plants. Radicle develops into root system, plumule develops into shoot system, and so on. That means the cells which are present in the body of organism, those are developed from the initial cell, pre-existing cell. And along with this third statement, the cell theory is now called as the modern cell theory. Now the question is that whether all the cells, they are of the same kind. We can see a wide diversity in the life forms. We have organisms like as a pleuropneumonia like organisms. They can change their shape. They are never stable in their forms. The archaebacteria, cyanobacteria, which are filamentous or unicellular, the algae, fungi, various kinds of plants, and animals. So these are broad groups of the organisms. But again, the question is same, whether all these life forms are made up of one kind of cell. Now, how we can divide these organisms in a specific group? We can divide the organisms based on the organization of the cell. So initially, we must segregate the cells in a specific group. Now, there are various characteristics which can be considered for the classification of the cell. Say, for example, the shape, then their size. But these characters, they do not show a particular continuity in the phase. Say, for example, the size of cell may varies from few microns up to few centimeters. There is a wide range of shapes in the cell. So these characters are useless as far as the classification of cell is considered. The cells can be divided on the basis of the organization of the nucleus. When we consider this characteristic specifically for the classification of the cells, we get only two groups. Either the nucleus is properly organized or the nucleus is not organized. So on this basis, we have two groups of the cell, prokaryotic cell. And the second group of the cell is called as a eukaryotic cell. In today's lecture, we will discuss the organization of prokaryotic cell. The term prokaryotic is derived from a Greek word. Pro means primitive and the karyon, which means nucleus. So these are the cells having a primitive type of nucleus. Now what is meant by that? The primitive type of nucleus, that means the nucleus without any well organized structure. So true nucleus is absent. The best examples are archaebacteria, eubacteria, and the cyanobacteria. In true bacteria or in eubacteria, we can take an example of Escherichia coli, the colon bacteria, or Rhizobium, which is a root nodule bacteria. And in cyanobacteria, which are commonly called as a blue-green algae, BGA. So in plants also, we have a group of organisms which shows prokaryotic cells, specifically the Nostoc and Anabaena. Now let us discuss the organization of the prokaryotic cell. Now in this figure we can see that the cell, it has a specific structure. It appears to be a rod-shaped or rectangular shell. Now the major parts of the cell includes which are named here, but we will start with the outermost membrane. So outermost living membrane is a cell membrane. It is shown here by a thin line. So this is outermost living part of the cell. You know that the cell membrane is also called as a delimiting membrane. That means it separates the cell content from the external environment. It is the border which identifies where from the cell is started, which are the parts which are present inside the cell and what is present outside the cell. So we can say that this is a delimiting membrane, cell membrane. This is made up of the lipids and proteins, and it is selectively permeable in nature. In addition, the prokaryotic cells are covered by a layer, specific layer, which is called as a cell wall. Now, the cell wall may be present or may not be present. A cell wall, in case of bacteria, these are formed of muramic acid. So they have muramic acid and peptidoglycans in their structural organization. Whereas in case of plants, the cell wall is made up of cellulose. The cell wall is externally covered by another layer, capsule, but again the capsule may be present or may not be present. Now this outermost layer, when it is present in form of a solid layer, it is called as a capsule, but sometimes it appears to be a slimy one, which is called as a slime. It is just like as gel-like structure. The capsule and cell wall provides additional protection to the prokaryotic cells. So in many cases, the prokaryotic organism which has a cell wall, they appears to be resistant to number of antibiotics. Whereas the cells without cell wall found to be susceptible to number of antibiotics. So this cell wall provides protection against the external abiotic factors like as a temperature, heat, or the antibiotics. The another role of cell wall is that this cell wall provides a specific shape to the prokaryotic cell. So depending on the shape, we have three types of prokaryotic cells. The bacteria, we have cocci, that means which appears to be spherical. Then bacillus. So here the bacillus we have shown in this figure. The cells are rod shaped, and the third type is called as spirulum, where the cells appears to be spring like. Okay, so these three shapes are given by the structure of the cell wall. When there is higher salt concentration or osmotic concentration outside the cell, the bacterial cells, they lose their cytoplasmic water and the plasma membrane shrinks. It is called as a plasmolysis. But even in the plasmolyzed condition, the cell does not change its shape if the cell has its specific cell wall. Otherwise, the cell will either shrunk or it will burst open due to endoosmosis. So cell wall provides protection. Before going into details of the internal structure, let us see what are the outgrowths of the cell. Now here we can see the flagellum is there. So flagellum are long whip-shaped structures which helps in locomotion of the bacterial cell, prokaryotic cell. The flagella may be present or may not be present. So we have marked the asterisks against those characteristics which may be present or may not be present. So flagella may be absent. Now when the flagella is present, the number of flagella may be 1, just like as we have shown here in this figure, or the flagella may be 2, or the flagella may be many. In addition, the cell membrane shows certain small outgrowths which are called as a pili or fimbriae. So these pili or fimbriae are present on the surface of the cell, prokaryotic cell, and they helps in the attachment with the substratum or attachment with the other cell during sexual reproduction. So this outgrowth, which is shorter as compared to flagellum, is called as villi, or it is called as fimbriae. Now coming towards the cytoplasm or protoplasm. So whatever living matrix is present, the life supporting matrix is present inside the plasma membrane is called as a cytoplasm. But again the confusion is that the cytoplasm, when it is separated from nucleoplasm, then we have two compartments. Now in case of bacteria, there is no nucleus, so nucleoplasm is not there. So cytoplasm number of times may be called as a protoplasm. Okay, so protoplasm is life supporting matrix. Now this life supporting matrix shows the important structure, that is nucleoid, the genetic material. Protoplasm shows some prominent characteristics. The first and important characteristic is that there is no compartmentation. That means whatever contents are present in the cell, prokaryotic cell, those are all present in common matrix. So there is no separate compartmentation like as a compartmentation for nucleus, compartmentation for chloroplast, photosynthesis, compartmentation for respiration. Nothing, no separation is there. So all these metabolic activities, these are carried out in the one and same cytoplasm. So we called them as a protoplasm. So no compartmentation is the important characteristic of prokaryotic cell. Now how the cell completes its function. Now we can compare the prokaryotic cell with the primitive type of school. So in earlier days, when I was studying in primary school, then our first and second standards, all the students were sitting in the same room. There was one teacher who was teaching us mathematics, who teaching biology, who teaching languages. So for different subjects, there were no different teachers. One same teacher was teaching us all the subjects. The same teacher was working as a principal or the headmaster. The same principal was working as a clerk. The same principal was working as a peon. So everything was there. All the functions were there, but there was no specification. Who will do which specific function, or who will be present at which particular place. So spatial separation is not there. Similarly, the functional separation is also not there. We can compare this condition with the prokaryotic cell. So here in this cytoplasm, there is no separate site for photosynthesis, separate site for protein synthesis, or separate site for respiration. All the functions are going on, but these are going on in the cytoplasm. Now let us see the structure of genetic material. The genetic material is DNA, deoxyribonucleic acid, like as that is present in the eukaryotic cell. But the specialty is that this DNA is usually a circular DNA, circular double-stranded DNA, which replicates itself. So this is called as a bacterial chromosome, B chromosome. And this B chromosome is circular, so it doesn't shows linear structure. It doesn't shows ends where the chromosome starts and where it ends. And it folds many times upon itself and it forms a complex structure. This complex structure is called as a nucleoid. Note another thing, that this genetic material is not covered by any other spatial membrane. So nuclear membrane is absent. So this nucleoid, the DNA material, may be present here in the cytoplasm in this position, at this position, or at any position. So this is said to be a primitive nature of the nucleus, and it is called as a nucleoid, or it is also called as incipient nucleus. Now in addition to this bacterial chromosome, B chromosome, sometimes the cell may carry additional genetic material, additional DNA, and this additional DNA is called as a plasmid. So plasmid is nothing but extra chromosomal, circular, double-stranded, self-replicating DNA. So again, this DNA is self-replicating, circular, having specific genes. The plasmids may be present, may not be present, just like as a capsule. It may be present, may not be present. Whenever it is present, it confers some specific ability to the cell. Say, for example, certain plasmids, like as F plasmid, will help to promote the sexual reproduction. It helps in conjugation. It helps in fertility of the bacterial cell. The R plasmids are there, which helps in providing additional resistance to antibiotics. So in this way, whenever plasmids are present, they will confer some additional characteristic, additional ability, additional power to the prokaryotic cell. The another cell organelle is a ribosome. Ribosomes helps in protein synthesis, and proteins are essential for the enzymes which carries out all the activities in living organism. So these enzymes are synthesized by a specialized assembly which is called as a ribosome. In case of prokaryotic cells, these ribosomes are of 70S type. 70 Swedberg's unit type of the ribosomes are there. 70S ribosomes are also called as a prokaryotic ribosomes. These ribosomes usually are dissociated in the cytoplasm. So smaller subunit and larger subunit will be separate. But whenever there is a protein synthesis, these subunits will assemble together on a messenger RNA and they will form a polyribosome. Then at certain places we can see here that the cell membrane is folded upon itself. These foldings may be present at one place, two place, or at many places. And these foldings are called as a mesosomes. Mesosomes helps in increasing the surface area. It increases the surface area of the plasma membrane within cytoplasm. And in this way, it provides the extended surface area for metabolic activities. The mesosome also helps in formation of the cell plate during cell division. So whenever there is a fission of the cell divides into two. The cell plate within these two daughter cells is contributed by the mesosome. The respiration is carried out by some respiratory enzymes, and these respiratory enzymes are present along the wall plasma membrane. This is called as a respiratory chain. Now note that the respiratory chain doesn't have any specific separate enclosure, separate membrane. Similarly, the ribosomes, these are the particles, they do not have membrane. The nucleus does not have membrane, and that is why there is no compartmentation. Now, in addition, the cell consists of number of inclusions. The reserve food materials may be present in the form of starch, in the form of protein molecules, in the form of oil droplets. So, depending on the type of bacteria, it has a specific type of reserve food material. So, this is a general outline of prokaryotic cell. The cell without well-organized nucleus, the cell without any compartmentation, the cell having 70S type of ribosome is called as a prokaryotic cell. So this is general organization of the prokaryotic cell. We can enlist the characteristics that the true nucleus is absent, incipient nucleus is present, the genetic material is scattered in the protoplasm, it doesn't have any distinct membrane. This genetic material is called as a nucleoid. The cytoplasm does not show any cell organelle. Membrane-bound cell organelle is absent. Compartmentation is absent. The ribosomes are primitive ribosomes called as 70S ribosomes. The cells are without compartmentation. Neither the nuclear material or the respiratory enzymes are separated by any membrane. And the best examples for the botany students are the blue-green algae Cyanobacteria, like as a Nostoc, Anabaena. True bacteria like as a Rhizobium, root nodule bacteria which are present in dicots, leguminous plants, Azotobacter, Streptococcus, etc. Hope this lecture was useful for you to understand the structure of prokaryotic cell. In the next video, we will discuss the eukaryotic cell and how the compartmentation and various cell organelles are present there. Till then, take care. Thank you.